Hello. In this Softchalk lesson, I'm going to try to explain a little bit more about how you can use your detailed learning objectives to make a study guide for each Softchalk lesson. As I mentioned in the very beginning of the semester and in the Getting Started videos and in a couple of announcements, your exams come directly from the detailed learning objectives found at the beginning of each Softchalk lesson. So in terms of having a study guide, what you need to be able to do is to go through and answer the objectives and learn those objectives. And that will give you the information you need for a study guide for preparing for that unit exam. So first of all, if we look at our Softchalk lesson here on the screen, I'm going to talk today about horizontal gene transfer in bacteria, your first lecture that was in Unit 2. And of course, as you know, if we look at our table of contents here for horizontal gene transfer and click on bullet points and objectives, we see the detailed learning objectives that you need to know on horizontal gene transfer for the Unit 2 exam. So there's a couple ways you can go about this. If you're old school, you can copy all of these down onto a note card and then go through and read each Softchalk lesson. And as you go through and read it, answer the objectives. Make sure you look at illustrations and animations and make note of any of those that might be useful in helping you answer the objective. It becomes very important to be able to visualize a process as well as just memorize the words. So if you can visualize it and you know the words, you can link that together and remember it better. So another approach, if you're not doing the note card, is if you're more used to using computers, you can just copy all those objectives and then paste those into a document that you can use to prepare your hard copy of your study guide. And you can also, of course, store that on your computer. So for example, if we cut and pasted these, let me show you where I've done that. Show you the screen with the objectives that you need to know for horizontal gene transfer right here. So here's the objectives you need to know for Unit 2, Lecture 1 on horizontal gene transfer. Now, first, as I mentioned in the Getting Started videos, take a look at your objectives. Notice that some of them have a single asterisk, some of them have double asterisks, some have no asterisks. And that gives you an idea of how much depth you have to know. If we have an objective like numbers 3 and 4 here, or parts B and C of objective 5, notice there's no asterisks. So that means that's something we need to know for the exam, but we don't have to worry about that too much for the rest of the semester. It's part of learning about horizontal gene transfer, but nothing we're going to really apply too much later on. And so what you'll probably see on objectives like 3 and 4, or parts B and C of objective 5, is a multiple choice question where you would recognize the description of that mechanism on a multiple choice question on the exam. If there's a single asterisk, like in numbers 1 and 2 and 6, that means it's a common theme throughout the course. So again, we need to know that for the Unit 2 exam in this case, but we'll also have to know that the rest of the semester. We're going to be applying horizontal gene transfer and especially conjugation, as we see number 5A, throughout the rest of the semester, or our plasmids in objective 6. And if there's 2 asterisks, we're going to have to know that in more detail. Like 5A says transfer of conjugative plasmids, conjugative transposons, mobilizable plasmids, and gram-negative. So there probably will be several questions on that. These are processes we need to understand. And so we need to know a little more detail about the process. You'll probably see several questions on that on the exam. So again, we need to know that in a little more detail. So once you have your objectives down, then you're ready to start answering them and preparing your study guide. So let's take a look at objectives 1 and 2. Now, objective 1 says compare and contrast mutation and horizontal gene transfer as methods of enabling bacteria to respond to selective pressures and adapt to new environments. So first we want to compare and contrast mutation and horizontal gene transfer. And then number 2, little more on horizontal gene transfer, define horizontal gene transfer, and state the most common form of horizontal gene transfer in bacteria. So now that we see those objectives we want to answer, let me bring up the screen for your Softchalk lesson. And we see that the first topic in horizontal gene transfer in bacteria Softchalk lesson is mutation and horizontal gene transfer. So there we're going to find the answers to objectives 1 and 2. If we click on that, so here we have objective 1. It says compare and contrast mutation and horizontal gene transfer. And here we see our answer to that. Mutation, where you're modifying a gene function within a bacterium. Horizontal gene transfer, where a bacterium is acquiring several genes from another bacterium. And we also see, and keep in mind that boldface means is probably important, mutations occur relatively slowly, and most mutations are actually harmful. But again, bacteria are so numerous that even though something is a rare event and most are harmful, you do get some mutations that provide a selective advantage and allow the bacteria to adapt to the environment. So whereas mutation is an important means of bacteria adapting to their environment, it is a slower process. Whereas horizontal gene transfer, as we see down here, enables bacteria to respond and adapt to their environment much more rapidly because they're acquiring several genes all at once from another bacterium, not modifying a single gene like in mutation. So that pretty much takes care of objective 1. And then if we scroll down a little, we have objective 2 on specifically horizontal gene transfer. So there's our definition for objective 2. And it tells you down here that the most common mechanism in bacteria is conjugation. So there's your answer for objective 2 on horizontal gene transfer. And you'd write those out. Now let's go back to our objectives and look at the next one. So we've just answered 1 and 2. We see number 3 is the only one that deals with transformation. Briefly describe the mechanism for transformation in bacteria. Again, no asterisks. So what you'll need to know primarily for the exam, there might be a single multiple choice question where you recognize the description of transformation and match that up with transformation. So to answer that one, if we go to our Softchalk lesson, let's bring that up again. Let's go back to our table of contents. So here we see the next topic, in fact, in our Softchalk lesson is transformation, objective 3. And there we have our description of transformation. And then there's a little more detailed description of how that process works. Now, again, since there's no asterisk after that, this is the basic description you need to know. But again, it's important to kind of understand the process, not just memorize the words, so that you can visualize it better and use that to help you answer the question. So if we go down a little bit further, we see there's an animation on transformation. So again, if there's illustrations or especially animations that deal with an objective, you want to make note of that when you're preparing your study guide so that you can remember to look at that visualization of the answer to that objective. If you can visualize what happens step by step during transformation, it makes it much easier to remember the description verbally. And so that answers your third objective on transformation. I should also mention that at the end of this particular video, I'm going to talk about the concept maps that are also available in the Softchalk lesson. And you'll see in the concept map, you also see the description of transformation as well as a link to the video, or in this case, the animation. And that will give you a nice sheet to use to review materials and help study. And we'll talk more about that later in the video. But anyway, there's our answer to transformation. and an animation that goes with that. So let's go to our next objective. Let's bring up our objective sheet again. Objective 4, briefly describe the following mechanisms of horizontal gene transfer in bacteria: generalized transduction and specialized transduction. Now, you may remember that you had the words transformation, transduction, and conjugation as part of your biology review exam earlier in the semester, and had some at least one question on each one of those. And I introduced those words early in your biology review since that was something we're going to be applying during the semester. But here again, we're getting into it where you really need to know it for the exam. So notice on objective 4, again, no asterisks. So basically you'd expect to see maybe a multiple choice description of generalized transduction and or specialized transduction, and be able to match the description with the type of transduction. So to answer objective 4, let's go to our Softchalk lesson and look in the table of contents for transduction. Okay, if we go back to our table of contents, we see that the next topic, in fact, after transformation is transduction. Now, first of all, as you might remember from your take-home exam, this is our general description of transduction in general. So whereas transformation was transfer of DNA from a dead, degraded bacterium to a competent recipient bacterium, transduction is where a DNA fragment goes from one bacterium to another, but it's carried by a virus called a bacteriophage, a virus that only infects bacteria. And here is our general description for generalized transduction. And as you go through and read that, you'll see that during generalized transduction, this is a mistake that occurs as the virus replicates, whereas the virus assembles within the bacterium. Occasionally, a single virus will pick up a fragment of bacterial DNA rather than viral DNA and carry that bacterial DNA from one bacterium to another. It can also pick up a plasmid and carry that with generalized transduction. So there we have our description of generalized transduction we need to recognize. And there's our animation you can go through. So again, you can visualize the process of generalized transduction, see how the virus picks up a little bit of bacterial DNA and how it carries that bacterial DNA to another bacterium. And then scroll down a little bit further and we see our description of specialized transduction, where in this case, it's a mistake that occurs during spontaneous induction, where occasionally a viral genome leaves behind a little bit of its DNA and by mistake picks up a piece of bacterial DNA. And then in this case, every virus is going to carry that little bit of bacteria DNA to another bacterium. And there's our description to answer the objective, and there's the animation where again you can visualize the process and how each virus picks up a little bit of bacterial DNA. And that pretty much will take care of transduction objective. Let's move on to our next objective. Okay, we have objective 4. We just did. Objective 5 says briefly describe the following mechanisms of horizontal gene transfer in bacteria. Now B and C have no asterisks. So as we go through there and we see F plus conjugation in the lesson and HFR conjugation in the lesson, we'll need to be able to recognize the general process on maybe a single multiple choice question. But notice objective 5A has 2 asterisks. So in terms of horizontal gene transfer, you need to describe how there's a transfer of conjugative plasmids, conjugative transposons, and mobilizable plasmids in gram-negative bacteria. So that one we're going to have to know in a little more detail. Also notice objective 6 also deals with conjugation, in this case, our plasmids. And again, there's a single asterisk, so we need to know a little bit more detail about that, and we'll have to understand that through the rest of the semester. So conjugation in 5A and objective 6 become important because this is the primary way that drug resistance is transferred between bacteria, transferred from one bacterium to another. This is also a way that virulence factors, the ability to colonize or the ability to harm the host, can be transferred from one bacterium to another. And because conjugation is rather promiscuous compared to transduction and transformation, this can be transferred to other unrelated bacteria. It doesn't even have to be the same genus and species of bacterium. And in the conjugation, we're going to specialize on how it works in gram-negatives, which is going to involve a conjugation pilus. So let's take a look at the answers for objectives 5 and 6, dealing with conjugation. So if we go to our Softchalk lesson, here's conjugation. We click on that. And there's our general description of conjugation in general. So remember, this is a transfer of DNA from a living donor bacterium to a living recipient bacteria by cell-to-cell contact. And in gram-negatives, it's going to involve a conjugation or sex pilus. So this is where a mating pair is going to form, where DNA can directly go from one bacterium to another. Unlike transformation, where there was a fragment of DNA from a dead, degraded bacterium entering a competent recipient bacterium, or transduction, where DNA is transferred from one bacterium to another by a virus or bacteriophage. So this is going to involve a donor that either has a conjugative plasmid or conjugative transposon. And these are terms we had back in Unit 1 under the nucleoid, under plasmids and transposons, where we defined conjugative plasmid, mobilizable plasmids, and down here transposons. But these definitions are repeated again in this unit in case you don't remember them. So in this case, there's going to be a conjugative plasmid or conjugative transposon that contains what are called TRA genes or transfer genes, and that's what allows bacteria to form mating pairs to carry out conjugation. And they also have a sequence of DNA called an ORIT, or origin of transfer sequence, that determines where a DNA strand can be cut and be transferred to a recipient. And again, here's where the animations have become real useful. If you just memorize those words, it's a lot of words. If you can visualize the process, it's pretty straightforward. So again, remember our objective 5 said we're going to look at conjugation in gram-negative bacteria, which is going to involve a conjugation pilus. So this gives you the background you need to know about conjugation involving plasmids and transposons, including mobilizable plasmids. If we go down now, scroll down to conjugation in gram-negatives, here's where we can answer objectives 5 and 6. So in gram-negatives, it's going to involve a conjugation pilus, and here's your basic description of the process. And once the mating pair forms, then one strand of a plasmid is going to leave the donor bacterium and enter the recipient. And then each bacterium is going to make a complementary copy of the strand that it has, and both bacteria will now have the conjugative plasmid. Now, again, that describes it, but if we look at the animation, we really need to be able to follow what happens step by step. If you can visualize this, it's much easier to understand than just memorizing a lot of words. So here is conjugation in a gram-negative bacteria. Notice this bacterium, our donor, has a chromosome, but no chromosomal DNA is transferred during this type of conjugation. This involves a conjugative plasmid. So notice that in addition to a chromosome, this bacterium has a conjugative plasmid. And as part of that conjugative plasmid, what makes it a conjugative plasmid is it has these transfer or TRA genes coded, shown in green here. And this is what codes for the ability to make a conjugation pilus in a gram-negative and form a mating pair in both gram-positive and gram-negative bacteria. There's also an origin of transfer area here. This is where a nuclease is going to split one strand of the plasmid so it can enter the recipient. Now, there may be other genes as well coding for various things. For example, if this was an R plasmid, that's a conjugative plasmid where these accessory genes shown in red code for multiple antibiotic resistance. But again, those could code for other factors as well. Virulence factors that allow a bacterium to be more harmful, or other factors that allow it to adapt better to the particular environment it finds itself. So because this bacterium has a conjugative plasmid, it's able to form a conjugation pilus if it's a gram-negative. And that pilus makes contact. Remember, pili are organelles of adhesion. So that allows the bacterium to make contact with the recipient. And then the pilus depolymerizes or retracts, pulling the two bacteria together. And then the TRA genes are going to code for some coupling proteins that allow the bacteria to form a mating pair. And that creates a channel between the two bacteria through which a strand of a plasmid could be transferred. So what happens next, now that it's formed a mating pair, is that a nuclease is going to split one strand of the plasmid where those gray ORIT genes are, and one strand of the plasmid is going to enter the recipient. One strand is going to stay behind. So here we see the outer strand entering the recipient, the inner strand staying behind. Both strands, by complementary base pairing, adenine pairing with thymine, guanine pairing with cytosine, are going to make complementary copies of themselves. And so when all is said and done, both bacteria now have the conjugative plasmid, and both bacteria can now conjugate with other bacteria because the recipient now has the TRA genes that allow it to conjugate, and the donor still has the TRA genes allowing it to conjugate. And of course, both now carry whatever these red genes code for. If this was an R plasmid, again, those red genes would code for multiple antibiotic resistance. But again, conjugative plasmids could have other genes that they're transferring besides drug resistance. Now, another thing that can happen, as long as the donor bacterium has a conjugative plasmid, it could also have a conjugative transposon, which is a jumping gene that codes for mating pair formation, not shown here. But what can happen is that bacteria may carry several plasmids. And some of these other plasmids can also be transferred during conjugation. And these are called mobilizable plasmids. Now notice a mobilizable plasmid has no TRA genes, none of these green genes. This can't code for a mating pair. So if this bacterium only had the mobilizable plasmid, but didn't have the conjugative plasmid, then it would then it couldn't form a mating pair. There would be no transfer. But because this bacterium has two plasmids, and one is a conjugative plasmid with TRA genes, this bacterium can form a mating pair and can conjugate with the recipient and form a mating pair with that. But what can happen now is that once a mating pair forms because of the conjugative plasmid here, is that a nuclease can also split a mobilizable plasmid, and a strand of that can go through the channel created by the mating pair. So as long as one bacterium has TRA genes, either in a conjugative plasmid or a conjugative transposon, and can form a mating pair, other plasmids can be co-transferred. Now, in this case, only a strand of the mobilizable plasmid was transferred, and then both strands make a complementary copy of themselves. So when we're done here, this bacterium now has the mobilizable plasmid and whatever that codes for, but it won't be able to make a mating pair because it didn't get a strand of this one. Now, it's possible that it could get a strand from both plasmids, but in this case, only the mobilizable plasmid was transferred. But this one still has a conjugative plasmid, so the donor can still act as a donor and form a mating pair. The recipient can't. And that is how conjugative plasmids and mobilizable plasmids are transferred by conjugation gram-negatives. So again, that one has two asterisks. You want to look at the description carefully. You want to look at the animation carefully so you can follow the process. And then for objective 6, In some bacteria, the conjugative plasmid is actually an R plasmid, which is the next topic down. And it's the same process as this, except now we're saying that these red genes in the conjugative plasmid are coding for multiple antibiotic resistance, as well as formation of a mating pair. So if this was an R plasmid, the process would be the same as we saw above, where because of the TRA genes, the bacterium can form a conjugation pilus if it's gram-negative. That can conjugate with a recipient. The pilus retracts, pulls the two bacteria together. The TRA genes code for the coupling proteins allowed to form a mating pair. The nuclease cuts one strand of the R plasmid at the ORIT section. That strand enters a recipient. One strand stays behind. Both strands make a complementary copy of themselves. And now both bacteria have an R plasmid. Both bacteria, because they have these red resistance genes, code for multiple antibiotic resistance. But both bacteria now have TRA genes, so both the donor and the recipient can now conjugate with other bacteria and spread R plasmids to them. So in R plasmid transfer conjugation, we're seeing the transfer of not only multiple antibiotic drug resistance, but also the ability to conjugate and spread that resistance to yet other bacteria. And again, because conjugation is rather promiscuous in bacteria, the donor can transfer plasmids to unrelated bacteria. It doesn't have to be the same genus and species. And that's one of the major reasons why bacteria can adapt so rapidly, because they can transfer these conjugative plasmids as well as mobilizable plasmids. And then parts B and C of Objective 5. Remember, they didn't have an asterisk beside them, F plus conjugation and HFR conjugation. So again, we just need to know a general description. So there's our general description of F plus conjugation. And in this case, the only thing that's transferred is an F plus conjugation, which codes only, or an F plus plasmid, which codes only for the ability to form a mating pair, nothing else. So that wouldn't seem to be too useful, but the point is that as long as the donor has an F plus plasmid and can conjugate, mobilizable plasmids could also be transferred. But anyway, there's our description we need to recognize, and there is an animation that shows how just an F plus plasmid coding just for mating pair formation can be transferred from a donor to recipient. And then we have our description of HFR conjugation. And this is the only case where there's a transfer of chromosomal DNA, but usually not the ability to form a sex pilus. And so what happens here is the F plus plasmid inserts into the chromosome. And then when the nuclease breaks the chromosome, one strand of the chromosome begins to enter the recipient, but usually only a bit of that gets through before the bacteria break apart. So they usually don't transfer the ability to make a mating pair, but they are transferring some chromosomal DNA. But anyway, there's our description we need to recognize of HFR conjugation. And there's an animation you can look at to follow the process so you can visualize it. So again, once you look at the objective, go to the table of contents of the Softchalk lesson and see where we discuss that objective. Read through the Softchalk lesson on that particular topic, and then answer the objectives and check out any illustrations or animations that go with that so that you can use that to visualize the process. Now, another thing you notice on the exams are the critical thinking questions. That's one of the things, especially nursing has asked us to do, is to have more critical thinking in the course to prepare people for nursing. And I do that with the think, pair, share questions that are mentioned there. So as you're going through and answering your objectives, you'll notice the think, pair, share questions are also listed there. And the key here again is that you can't really answer these think, pair, share questions until you've studied the other objectives, until you know the content, because these test your ability to apply what you've learned. If you haven't learned it yet, you can't apply it. But what you want to do is simply look for keywords. if you've studied the material, it's usually pretty easy to find the answers. So if we look at our first think, pair, share question, first of all, it says there's a strain of living Streptococcus pneumoniae that can't make a capsule and therefore is harmless. Without the capsule, the bacteria can be phagocytosed and they aren't harmful. So a living bacterium that can't make a capsule is mixed with a killed sample of bacteria that could make a capsule when it was alive. And after a period of time when that mixture is injected into mice, they're killed because they can now make a capsule. So it's asking you for, in terms of horizontal gene transfer, describe what accounts for that. Well, again, look at keywords. We have a living bacterium being mixed with a killed bacterium. So where's the only place where we saw a transfer of DNA from a living donor bacterium to a from a dead degraded bacterium, I should say, to a living recipient? And that was transformation. Remember our definition, transformation is DNA from a dead degraded bacterium, and there's a competent living bacterium, and that DNA can then be exchanged. So that would describe transformation. If we look at number 2, we see there's a gram-negative bacteria that was susceptible to most antibiotics, now is becoming resistant to several antibiotics, and it seems to be spreading that resistance to other bacteria. So again, we look at our key words. There's a bacterium that's now multiple antibiotic resistant, transferring that resistance to other bacteria. So again, we look at what we just studied, and the only place where we described multiple antibiotic resistance and the ability to form a mating pair and conjugate and spread that to other bacteria was our plasmid conjugation. So that's a description of our plasmid conjugation. Now, I also want to point out finally at the end here that remember, for each Softchalk lesson, there is a concept map that summarizes all the key points and gives you a lot of the information that's an answer to your objectives. Now, I didn't include the little blurb on mutation here since that was something you had coming in, but this deals with the whole horizontal gene transfer part. So remember, objectives 1 and 2 ask you to compare and contrast mutation and horizontal gene transfer. The mutation part's not here, but again, here's our description of horizontal gene transfer and what it does, the benefit. So that answers objective 2 and part of objective 1. Objective 3 was transformation. So one of the mechanisms of horizontal gene transfer is transformation. There's the description we need to recognize, but also if we click on the little box here, we see that that takes us to the animation for transformation you can use to review the process and visualize it. Another mechanism of horizontal gene transfer, which is objective 4, was transduction, where viruses are transferring DNA from one bacterium to another. There are 2 types, generalized and specialized. So there's our description of generalized transduction that we need to recognize, and a link to the animation of generalized transduction so you can visualize that process. And here's our description of specialized transduction, along with a link to that animation. And then the third mechanism of horizontal gene transfer is conjugation. So there's our description in general of what conjugation is. And that can involve conjugative plasmids, which are described here, mobilizable plasmids, which are described here, conjugative transposons, which are described here. And it can also involve F+ conjugation. So there's our description of F+ conjugation, which was objective 5B, and the animation that goes with that description. Objective 5C was HFR conjugation. There's the description we need to recognize. There's the animation that allows you to visualize it. And then objective 5A, the one we have to know in detail, is transfer of conjugative plasmids. So there's the description, what the TRA genes do. And there is our animation for transfer of a conjugative plasmid that we went through. And sometimes those conjugative plasmids are our plasmids that code for multiple antibiotic resistance, as well as the ability to form a mating pair. So there's a description of our plasmids and the animation that describes that. So everything we put down in our study guide there that we prepared from the detailed learning objectives, a lot of that is summarized also in your concept map at the end, along with direct links to animations and illustrations. So don't forget the concept maps. They can be very useful to you in helping to review this once you've learned the material. So anyway, that is a little video on how to prepare a study guide based on your detailed learning objectives found at the beginning of each Softchalk lesson. Write out the objectives or copy them, and then go through page by page of the Softchalk lesson. You can look in the table of contents and get a pretty good idea of where that objective is answered. Read through that section, such as transformation, and then answer the objective and take note of any animations or illustrations that might be useful to help you visualize the process. Once you're done, you have pretty much the material you need to know for the exam. And then don't forget to look at think, pair, share questions. Once you've studied the information in that Softchalk lesson and understand it fairly well, then look for keywords in the think, pair, share question and see where we discussed a topic that has those keywords and see if that helps you answer the think, pair, share question. And then to continually review the material, don't forget to look at concept maps where they'll give you a lot of the key definitions and descriptions, as well as direct links to animations and illustrations to help you review material. So once you've prepared that study guide, then you're well on the way to what you need to learn. Then it's a matter of putting in the time to learn those objectives. So again, once we go back here to our objectives, once you've answered them, have them written out, you can use that as well as the concept maps to keep going over that. And a lot of that gives a repetition, going over it till you can understand it. So you want to get where you can read yourself an objective and then answer it in your mind. What's the answer to this objective? And don't forget, look at illustrations, animations, so as you're answering, you can visualize the process of what's occurring. And again, that's where the time comes in. As we've said many times now, especially in a blended course, that adds time because you're not coming to lecture. So that's also part of the time it takes to learn the material. You should be spending probably a minimum of 12 to 15 hours a week to really understand the material that we covered that week. And again, this is important to realize the time commitment. One of the things we have to do legally in our syllabus is have a section that we've pointed out several times on out-of-time class requirements. And there was a section that indicates that for a 4-credit course, it would take a minimum maybe 8 to 12 hours a week, additional 3 hours if it's a blended course where the lecture is online to learn the material, and that might be a minimum. And the reason that is put in there by the federal government is that many students are receiving federal money to attend college, and they want the students to realize that taking a college course is a time commitment, that if they're getting the money for this, they have to put in a significant amount of time to understand the material they're learning. So again, that is a requirement of the college that we put that in. And again, a reminder that it does take a lot of work. But again, the material is well organized. You have lots of illustrations, animations, and other links. You have self-quizzes along the way you kind of use to see if you're understanding some of the basic terminology at least. And you prepare a study guide using your detailed learning objectives. And most people who do this do very well in the course. So I hope you found that useful. If you need more help with this, feel free to see me during my face-to-face office hours or to make a calendar appointment from your announcements to set up a slot of time where you can have a Zoom office hour. Again, if you're studying materials and you've tried to learn it and you're having trouble with a think, pair, share question, you can always make an appointment to go over some of that. But hopefully you'll find this somewhat helpful, and I wish you good luck.